Okay, hi, I'm Mike Lawless, and I'm here today with Ray Chaudhuri of Janssen Pharmaceuticals. How you doing, Ray?
I'm doing good. Thanks for having me.
Very good. And we're going to talk about an article that was published in AAPS News Journal about a year ago, entitled Physiologically Based Pharmacokinetic Modeling and Simulations for Drug Candidate Optimization and Selection. So, can you tell me a little bit before we start what you do at Janssen?
Well, I'm a part of this organization called Discovery Sciences, which is predominantly engaged in discovering new chemical entities for different diseases and therapeutic areas. And my particular department is called Drug Metabolism and Pharmacokinetics. And what we do on a regular basis is support projects in these therapeutic areas as they're coming up with new molecules to take them from early discovery to late discovery, early development, and to all the way to the clinic.
The first section in the publication talked about predicting oral bioavailability in silico. Could you tell us a little bit about that study?
Well, we took roughly 60 molecules, and just based on their structure, we made in silico predictions of what their DMPK properties are going to be. For instance, pKa, log D, protein binding, oral permeability, etc. And using that in conjunction with a physiologically based pharmacokinetic model, we predicted what its bioavailability and exposure and plasma concentration time profile is going to be. The best part was that data for these 60 molecules was already out there. So we could compare the outcome of our predictions with the observed data.
Okay, very good. And how the predictions came out?
They actually came out to be pretty good. A majority of these compounds was within a twofold error of prediction, which is a very good benchmark when it comes to predicting exposure in the plasma, both for animals as well as clinic or human.
Excellent. Okay. And then the second section talked about Risperidone and modeling that. Can you discuss that?
As opposed to looking at an array of molecules and modeling them using a uniform cookie-cutter approach, we wanted to focus on Risperidone. And because it's a well-known molecule, over the years people have generated enough in vitro DMPK data for us to work with. But what we first did was take the structure of Risperidone and predict its DMPK properties in silico, and use that in conjunction with a PBPK model, just like we did with our first case study. And it turned out to be fairly well in terms of Cmax and AUC for the observed plasma concentration profile.
I see.
Excuse me. But because we had measured in vitro DMPK inputs, we thought we would do the same by entering it into the PBPK program. And as expected, it did improve the prediction. It improved the Cmax and the AUC. Now, the message here was that if you have measured in vitro DMPK inputs, by all means they should be used in conjunction with the PBPK model. But oftentimes when you're working in early discovery, you do not have access to such data. And this particular example shows how good or how bad in silico predictions would be in conjunction with PBPK as opposed to in vitro. So this was comparing in silico PBPK approach with an in vitro PBPK approach. And the in silico approach scored fairly well.
And Risperidone does some lysosomal trapping also. Can you explain what that is?
That is correct. That was
in the simulations.
That was a modeling and simulation curveball that we had to handle. So because of its high basic pKa and high log D, Risperidone is a perfect candidate for being trapped by lysosomes, especially when it enters the gut enterocytes, as well as the liver hepatocytes.
I see.
And we used a separate program called Membrane Plus to actually validate that. And once it was validated, then we incorporated that into GastroPlus. But because GastroPlus does not have a way to directly incorporate lysosomal trapping, we mimicked it through some kind of binding. Okay. In this case, enterocyte. So enterocyte binding, which is typically kept at 0%, 100% free, we optimized the enterocyte binding to reflect the lysosomal trapping that would actually happen mechanistically in vivo.
Okay, so less of the compound is escaping the enterocytes and going into the portal vein.
Actually, all of the compound would eventually do that because that binding would be reversible, but it
I see.
slows
Okay,
down
it
the
slows it
kinetics.
down.
Yes,
I
it
see.
slows down the kinetics well enough that that you capture the actual trend.
So then a final step in a drug is the formulation properties of it. Can you talk about how you use the modeling to enhance the formulation? Or
Sure, it's one of the common uses of PBPK and GastroPlus in the fact that whenever there is a change in formulation, either a new excipient added or a particle size change or a particle size distribution change, or going from a solution to a solid oral dosage form, the question is what would be the change in exposure because of the change in formulation. And such PBPK models are combined with mechanistic absorption models of the GI tract to assess the changes in such formulations on exposure without actually doing the experiments. It's
the in
routinely
vivo
done.
experiment.
Yes, in vivo, either in animal or in human.
I see.
And it's done fairly routinely.
Excellent. Well, thank you very much, Sil, for that explanation of this article. And remember that this article is available for download on the Simulations Plus website in the resources section.
Thank you. Thanks for having me. It was a very spirited discussion, and I'm glad we could talk about it.
Thanks, sir.